large challenges. In association with the Indian Pharmaceutical Association, South Tamil Nadu local branch, head office at Arulmigi Kalasalingam College of Pharmacy. Can you hear, sir? Can you hear, Professor? Can you hear?
Very well, yes.
Yeah, thank you, thank you. Today we are proud to have an eminent, distinguished personality, well-known academician, administrator, and honorable professor, Dr. Gerrit Borchard, president of Swiss Academy of Pharmaceutical Sciences, section of Pharmaceutical Sciences, Institute of Pharmaceutical Sciences of Western Switzerland, and Biopharmaceutical Sciences, University of Geneva, Switzerland. Sir, it is a great honor to have with us for this occasion, sir. We thank you for kindly being the valuable part of this session. Your prestigious presence is a great honor for us and great motivation and inspiration for our entire student community, sir. We fully appreciate you for finding the time to be with us despite of your busy schedule, sir. Welcome, Professor Gerrit Borchard. Welcome, sir.
Thank you.
Thank you, sir. Next, I would like to extend our sincere thanks to Professor Chinnachami, sir, IASC, President of IASCP, for his kind guidance and support in all our endeavors. Next, I would like to thank our special thanks to Dr. Manivannan, yes, Manivannan, Deputy Director of Drug Controller, India, our President, our President of IPA, Tamil Nadu. Thank you, sir, for a keen interest in every aspect of this institution. Thank you, sir. Thank you, Manivannan, sir. Very kind of your support. Next, I feel very much thanks to Dr. Jaiskilan, sir, and Satish, sir, for your wonderful arrangement. For your unconditional support and encouragement, make us move towards the achievement. Thank you, sir. Thank you, sir. Next, I render my heartiest welcome to my colleague. Faculty member and dear for today's session. All the very best, my dear students. Thank you, Gerrit Borchard. Thank you, sir. Welcome, sir. Please continue. Please continue. Professor Gerrit Borchard, please continue. Please continue.
Okay. Oh, I'm sorry, I missed that. Very sorry for this. So I try to share my screen. I hope you see my my presentation here.
Yes, sir.
Thank you.
Yes, sir.
And I'd like to extend my my thanks to the organizers. It's a big honor for me to be able to present today this international virtual learning series. You may or may not know that I have some connections to your state of Tamil Nadu. One of my former PhD students comes from your province, from your state. Dr. Shakti Kumar Raghupati, who did an excellent master's thesis and PhD thesis with me. And now, based on the technology he developed during his PhD thesis, he is now incorporating a startup company here in Geneva. And I'm very happy to be part of this experience. The other thing I'd like to thank you for is I have been to India like 6 times before, and this must have been my shortest commute until now. So thank you very much for making this possible, that I can be here in my office in Geneva and be able to present some of my thoughts on nanomedicine, which have a small scale. However, there are large challenges to be faced. So I set my thanks to you. This is actually the Lake of Geneva on the Lausanne side. But I'd like to also extend a thanks to all the workers, the medical doctors, the nurses, the pharmacists that have been working under these pandemic conditions. In fact, my wife is a pharmacist. She works in a public pharmacy. She has faced many of the problems that I'm sure you are facing as well. My older brother is a male nurse in a hospital, and he was also confronted with quite some severe conditions there. Our students, our graduate students, helped out during the last couple of months in the hospitals, in the pharmacies. So a big thank you to everybody who contributed to that. Now, I myself and one of my students, we spent 4 months in the lab here at Geneva in cooperation with the Chulalongkorn Hospital in Bangkok. We are on track to develop an mRNA vaccine against COVID-19, which goes now, as we speak, into the first animal trials, and we keep our fingers crossed that this will work out. Talking about the vaccines, you see here from the New York Times, that was drawn from the web yesterday. There's a coronavirus vaccine tracker, and you see that there are many 135 preclinical tests, among which are ours as well, phase 1, phase 2, phase 3. And I understand that in China, there's one that it has been approved for the use for the military there, without too many data being published at this point, but that gives some hope. Now, if we look at the different vectors that have been used for delivering this vaccine, you see there are live attenuated viruses, non-replicating viruses, but also virus-like particles, DNA and RNA. So more like advanced version of vaccines, which do not deliver the antigen itself, but more the genetic information for an antigen, which then would cause an immune response. Here you see clinical phase candidates, and again you see mRNA here, you see others that are being used in order to develop these vaccines, and there have been, as of April 9, in phase 1 studies, and we hope that some of it is working. Now what happens, this is vaccine development. We've been working on this for a long time already during my time at the University of Leiden in Holland. You see here the classical ones, like the live attenuated, whole inactivated, and so forth. But here you also see the DNA and RNA vectors that have been used. So we are basically, in my lab, we're currently focusing on these DNA RNA vaccines. What's the advantage of an mRNA vaccine? Well, as I said, you do not deliver the protein or the antigen itself or part of the pathogen inactivated and so forth, but only the genetic information. Now, and then the antigen is produced by the cell of the vaccinated person after the mRNA has gotten into the cytosol, reaches the ribosomes, where then you have this translation from the mRNA code into the peptide sequence. The advantage is that, in contrast to the DNA vaccines, RNA does not have to reach the nucleus in order to be transcribed, but only the ribosomes there. Now, there are different other advantages. You may reach a cellular and a humoral immune response, which means you have something like the immune system cells that are reacting, cytotoxic T cells, and so forth. I'm sorry, there's a noise there. Okay, I'm loudspeakers because there's a noise in the transmission. There's the humoral immune response. These are antibodies that have been produced and so forth. So advantages are here also stability during storage and transport. You have a fast production, and maybe you have seen that Elon Musk and his Tesla company, they're now building RNA microfactories for a certain company, CureVac, for mRNA vaccine production. So it's high tech. Now, here you see the concept of this. You see the protein that's been made from the transcription, translation from DNA to RNA to proteins. Afterwards, well, you get this information by sequencing the virus-specific information here, the genome of this virus. Then you transfer the mRNA to a certain plasmid vector, you administrate the vector, and voila, hopefully you have protection. Now, the thing is, this is the concept again. So where where you get a transfection with the mRNA, you get translation at the ribosomes. You have a protein that's either processed intracellularly and then presented by these MHC class I peptide, which then reacts with CD8 plus T cells. So the cytotoxic T cells, that's the cellular immune response. Or it's been secreted from the cells, the transfected cells, and then taken up again and then presented by MHC class II peptides to interact with the T helper cells. Now, in order to get there, I'm going back for once, you have to have some kind of carrier, some kind of vector that transports your highly charged RNA into the cells. Now, this is the vaccination side. Here you have a distinction in size of these vectors. So if you have something that's in between 10 and 200 nanometers for the vector systems, which transfer the RNA, there's a direct uptake into the lymphatic system, which then goes to the lymph nodes where this presentation of the antigen towards the T cells is happening. Or you have something here above 200 nanometers in size, you have a direct uptake by the dendritic cells, and then these dendritic cells migrate again through the lymphatic system and present them to the T cells. This is, again, the whole concept of this presentation of the antigens to the T cells. Now, this is complicated, right? We have been, as I said, we've been working on that formulation, also with the help of some companies, to develop a liposomal formulation of mRNA and DNA for certain antigens of COVID-19. We did the test in vitro here in our lab, and then we tech transferred the whole stuff over to our colleagues at Bangkok, Chulalongkorn, who are now going into mouse studies. And keep your fingers crossed that we can enter afterwards, after GMP production, which we may be doing here in Geneva as well, into clinical phase 1 studies and in parallel in vivo tests in non-human primates. Now, this has been probably a little complex. So speaking of complexity, as you see here, I don't have to tell you that nature is complex. You're scientists, you know, nature is complex. As is depicted, for instance, here in this nucleic acid sensing scheme of activation of dendritic cells to be activated against tumor cells that you find here. So how do you activate these dendritic cells that they attack cancer cells and destroy them? Now, you see a lot of regulation here, a lot of interactions. I, for myself, in my group, and this is Maria Petrovic, who is working on this, we are only looking at this little interaction here. So certain nucleotides that have been delivered to the dendritic cells and find their target, the receptor in the STING protein, which then activates through NF-kappa B the whole cascade of dendritic cell activation. So yes, this is complex. So what? We know that. Diseases that you want to treat, be it cancer, be it infectious diseases, are complex. And they're not only complex, but they're also dynamic, as you see from these bioinformatics data that I got out of a website here, HLEAP, who are working on bioinformatics. Here they have a bioinformatics analytics analysis of different pathways, of different molecules, of different receptors involved in a disease process. Now you see in the beginning of the infection or of the disease, there are many, many pathways, molecules involved. Now, during treatment, this changes, and it changes again after 3 months. And after 1 year, you're almost back to normal, let's say, so you don't have too much of an implication of different pathways anymore. So over the course of treatment, you have different dynamics and complex pathways which are interacting with each other, and that you have to take care of, right? And the treatment that I mentioned has to be precise, and it has to be rather personal. Now, this is not a dollar sign here. This is meant to be a DNA. For the majority of patients, you can use one treatment regimen, one dose, one combination of drugs. However, there are always outliers, like this Dutch lady here on her bike that has a different need for dosing, maybe a different need for treatment in terms of drugs or drug combinations that is different from the rest of the population. And there are others here as well. So if you want to treat, for instance, a cancer, you should be precise and you should be personal. Your therapy should be adapted to the personal expression of certain antigens of this person as well. Yeah, so what? If you only have a hammer, all problems are nails. However, we know from experience, yes, you may end up putting the screw into the wood here by using a hammer, but it's not the optimum solution. So not all problems are nails. We need an approach that takes into account this dynamic complexity that I just showed you of diseases. So how can we do this? What are the tools to do this? Well, as Swiss, we invented the Swiss knife. We have complex tools. We have to develop complex tools. So that means that our drugs are complex as well as the diseases we're looking at. Now, nanomedicine is one of these tools that you can use, complex tools. And in the following, I'd like to show you a little bit of the complexity of this nanomedicines. If you look at the history of development of treatment of diseases, before the 1950s, approximately, don't take it for the year, but in earlier times, let's say at the beginning of the last century, there was observational, traditional medicine, herbal medicines, and so forth. Then after the Second World War, 1950, there was a paradigm shift towards chemistry and biology. The synthetic drugs, receptor theory took hold, although it was already defined here in 1900 by Paul Ehrlich as well. And we looked at pharmacokinetics. So what is the exposure of a patient to a drug, and what does that give us in terms of efficacy? So all these drugs you see here were developed during that time. There was another paradigm shift with the development of the knowledge, of the scientific knowledge we have, with all the omics been invented and applied. So we have the human genetics, we have molecular biology, we have systems biology, we have systems pharmacology, where we can look at this complexity of disease processes. So coming from a traditional medicine through a chemistry and biology-based medicine, we're now at the pathology-directed medicine, so coming from an observational standpoint to a more molecular standpoint. And there is the part where nanomedicine now plays a role. So my own history with nanomedicine started at the University of Frankfurt, where I studied pharmaceutical sciences. I'm a pharmacist by training. It was in 1989, not much was happening. The Bengals led the charge with Eternal Flames. Indiana Jones was on his last crusade, and there was something going on with the wall in Berlin I don't remember anymore. Now, I spoke to this gentleman, Professor Kreuter, who was supposed to become my thesis supervisor, and he threw expressions at me that would become part of my thesis, like nanoparticles, complements, opsonization, expressions I never heard before during my time as an undergrad student at Frankfurt. So what I did, I scratched my head and I went to the library to look up these expressions. Oh, for the young people, a library is basically a Wikipedia, but offline. So we had these things on paper called books. Oh, never mind. So we started with this. And I said I am alumni of Frankfurt University, and I cannot talk about nanomedicine without mentioning Paul Ehrlich. Now, Paul Ehrlich was a researcher at Frankfurt University at the time, before 1900. He watched an opera in the Frankfurt Opera House called Der Freischütz. In this opera, there's this guy who wants to win the heart and the hand of the daughter of the head forester. And therefore, he takes part in a contest, in a shooting contest, and he acquires some magic bullets from the devil in exchange for his soul that always find their goal. No matter where he shoots at, he always finds their goal. So he wins the contest and the forester's daughter. Now, inspired by this, Paul Ehrlich created the concept of drug targeting. Vector systems, drugs that always find their target, no matter where you inject them, they always find their target. That was the idea of Paul Ehrlich, who received a Nobel in 1902, I think, for his work on some other stuff. Now, this tells you, if you know these sweets there, it's a typical Swiss sweet. So, wer hat's erfunden? Who invented it? And actually, nanomedicine has a long history in this country, in Switzerland. It was in the 1970s, which looked like this. Believe me, I lived through it, so I know. There was Professor Speiser, who unfortunately died in 2013, 7 years ago, who was professor of pharmaceutical technology at ETH in Zurich. Together with his two assistants, my supervisor at the time, my former professor in pharmaceutical technology at Frankfurt, these three together, they applied nanotechnology to drug and vaccine delivery already in the 1970s, 1976, 73. They already thought about using these nanomedicines in order to improve, maybe to drug target drugs using this nanotechnology. Now, how do you have to imagine this? I showed this before. You have to have some kind of vector. In this case, I took a liposome, which is like a double layer of phospholipids, much like the cell membranes. And you put a drug into it. In this case, this is doxorubicin. Then you can add some polymers on the outside, like polyethylene glycol, which makes it more hydrophilic. So the immune system will not recognize that right away. And you can put some targeting moieties, like these antibodies, on the surface. Now, that's the theory. Then you inject this into the bloodstream here. And then, this is a tumor tissue. The theory of this enhanced permeation and retention effect, or EPR, is that these particles can easily diffuse out of the vasculature into the tumor tissue, because in this tumor, the vasculature is rather leaky. There are large gaps in there, so it can get into the tumor. However, there's an absence of lymphatic drainage. So retention, these particles with their drug load are retained within the tumor tissue and then can have an effect. That's the famous EPR effect for solid tumors, created by Professor Maeda in Japan quite some time ago. But honestly, Paul ehrlich in German means honest. So honestly, Paul, where are we with this concept of drug targeting? Now, there was a recent review of literature looking at different studies done with nanoparticles. They looked at different publication years. They looked at inorganic or organic materials like polymers, like gold or iron oxide nanoparticles. They looked at passive or active targeting strategies, at size, at surface charge of these nanoparticles, everything else. But what they found overall is an efficient median of delivery of 0.7% of the initial dose. This is not so magic. If you think about this, these are not really magic bullets. That's a little higher than without a targeting approach, or just introducing the drug itself without a vector into the bloodstream. However, not so magic. If you look at other constructs like antibody drug conjugates, ADCs, the efficient median is a little bit higher, like between 1% and 2%, but still no magic. Now, what can we do about this? Or why is this? Here I show you, I'm professor in biopharmaceutics. It's always been a big interest of mine, because it shows you the interaction of drugs with the biological system they have to work in. Now you have this drug. This is for, Biopharmaceutics 101. You have a drug with certain physicochemical properties. With a formulation, you can improve on these properties, increase solubility, for instance, and then you can create an ADME profile, absorption, distribution, metabolization, elimination. And based on these ADME profile, you have effects because it interacts with the target, this drug, or you have secondary effects. This is true, and you can define this drug for the structure like aspirin, but also for more complicated structures. So you can pretty much determine this. However, when we come to pharmacist 2.0 here, or super pharmacist, it gets more complex. So if you're looking at these complex drugs here, nanoparticles, dendrimers, liposomes, and so forth, polymeric micelles, it gets more complicated. Sometimes you don't even know what the active pharmaceutical ingredient is, what the drug really is. So if we don't know what the drug is, how can we establish a ADME profile? Also, sometimes the target is not clear, and therefore, to make a connection between the drug and its physicochemical properties or the formulation with the effect or side effect is sometimes, or most of the time, really complicated. Another thing that plays a role for these complex drugs, because they are particles, they are recognized by the immune system as something like a bacteria or like a virus. There's an implication of the immune system, or it can lead to immunotoxicity, something you do not normally see for small drugs, right? Now, what is nanotechnology? I have a very good, in my opinion, definition by Dr. Raj Bava of the Albany College of Pharmaceutics in the US. He is a pharmacist and a lawyer at the same time. Nice combination. So what he said is the definition is the design of nanotechnologies, the design, characterization, production, application of certain structures and so forth by controlled manipulation of the size and the shape at the nanoscale, which then produces structures, devices, and systems which at least one new property characteristics that is superior to when you do not apply nanotechnology. Now, one of the key words here is size, right? So people have been talking about size for nanoparticles, nanomedicine for a long time. A lot of discussion on both sides of the Atlantic and in Japan and everywhere. Now, in 2017, December, FDA issued a draft guidance. This is not carved in stone. This is just a draft where they talk of nanoparticles or nanomaterials for use in pharmaceutics or in drugs of a scale of a size range between 1 and 1,000 nanometers. That's good because, most of our liposomal formulations, nanoparticles, are somewhere between 200 and 300. That's a scale that had not been recognized before. Before that, it went from 1 nanometer to 100 nanometers, and now it's all the way up to 1 micron, which is more realistic in my opinion. But again, not carved in stone, not a law yet. This is just a guidance by FDA and a suggestion by FDA, which makes sense in my opinion. CDER, that's the institute at FDA that receives the applications for new drugs, abbreviated new drug application, investigational new drug applications for nanomedicine. So these are applications that contain, in some sense, nanomaterials. And you see the curve is slowly going up here. So nanomedicine, after, 30, 40, 50 years of development, is not a science fiction anymore. We find current nanomedicines on the market here in Europe, as well as in the Indian market, I'm sure, coming from Embizome. Gilead Sciences is in the news with the remdesivir, of course. But in this case, they had a liposomal formulation of an antifungal amphotericin B. There is, for instance, this doxil I was talking about, that's liposomal doxorubicin. Abraxane is a combination of paclitaxel attached to albumin nanoparticles for the treatment of different cancer types right now. But I would like to focus on these iron carbohydrate drugs because we have done some research in my lab on this. This is iron sucrose in this case. There are many others, of course, but these are nanoparticles that are being used, as you can imagine, for the treatment of anemia. So you have a low hemoglobin count. These patients are given these drugs, iron carbohydrate drugs, in order to replenish their iron content to make new erythrocytes. Now, anemia, according to the WHO, is a global problem. I think it's one of the has the highest prevalence of food or diet-related diseases in the world. So you see there's an incidence or prevalence, population affected of 1.6 billion people related to different ages, to different diseases, chronic kidney disease, for instance, and other stuff, but also in pregnancy. Pregnancy is not a disease, of course, but not pregnant women are also affected here, as you see, by this low iron count in their blood. Now, how do they look like, these iron sucrose nanoparticles? Well, you have to imagine them like an M&M. You have a core of iron 3 oxyhydroxide core here, which is covered with a carbohydrate shell. The carbohydrate shell in this case is sucrose. They have a molecular weight, these particles, that you can measure by about 34 to 60 kilodalton, and a size, which I will show you, of about 8 to 10 nanometers. 8 to 10 nanometers only, very small. The problem here is that sometimes some of this iron leaks out from these nanoparticles through the carbohydrate shell and then is presented in the bloodstream. We call this labile iron, and it's supposed to create reactive oxygen species that can give you some side effects, potentially harmful inflammations in the kidney, for instance. So we think that, labile iron here, which gets out of the core from these nanoparticles, actually causes the side effects that we see. Now, the pharmacokinetics of these nanoparticles is clearly governed by interaction with the immune system. These particles have to be taken up by macrophages in order to be processed and then replenish the iron pool. And I'll show you here. So this is taken from a publication by Zeng et al. From 2017, where he tried to describe the pharmacokinetics of iron sucrose in this case. So these are infused into the blood circulation of these patients by an infusion bag. Then there are two things that are happening. One is they're degrading during the circulation, releasing some labile iron during the circulation there, degradation. On the other hand, they're recognized by the immune system, the reticular endothelial system, taken up by phagocytosis, and then further processed in order to replenish the storage of hemoglobin here. During the uptake here into these cells, there are two things that are happening as well. One is, of course, degradation of these particles in order to have the ferritin-bound iron here that is then going into storage. On the other hand, there's also some part of this iron that can leak out of these cells and can get into the serum as labile iron and do some damage there. In the serum, then the labile iron is bound by transferrin to transferrin-bound iron, which then also goes into storage. Now, as I said, this labile iron gives you side effects, inflammation, reactive oxygen species. Now, the phagocytosis here depends, the recognition of these nanoparticles by the immune system is based on, or is correlated to the surface properties of these nanoparticles and their size, for instance. So these are different parameters of these nanoparticles. We call them critical quality attributes, or CQAs, which then lead to the interaction with the immune system or the biological system. I talked about also the in vivo release. This is related to the degradation kinetics of these nanoparticles during circulation in the bloodstream. And the degradation kinetics, as I said, also play a role after phagocytosis by these macrophages there. So you could think now there's a large market for these drugs. What you can't think about is to make a Me Too product, a generic of these drugs. Easy, you only need iron hydroxide, particles, little nanoparticles, and coat them with a carbohydrate. Sounds relatively easy. That's what I thought in the beginning as well. I learned my lesson. Now, can you make generics out of these complex nanoparticles, out of these complex drugs? Now, you probably know that we have legislation for the approval of generic drugs. These are small molecules, molecular weight smaller than 500 Dalton. They can be fully characterized. You know their NMR spectrum, the UV spectrum, you know their molecular weight, no change, right? We have now for these biosimilars, follow-on products of therapeutic proteins, we have the biosimilar regulation in place. Since in Europe, 20 years, in the US, a little less than that. And there were no major incidents with these biosimilars. They're pretty safe. However, these are complex drugs. They are much bigger. And because of their complexity, microheterogeneity of these drugs, they cannot be fully characterized. And this is why we don't call them biogenerics, but biosimilars. Now, one could think if these are complex drugs of biological origin, can we do the same for nanomedicines of non-biological origin? Can we have something like nanosimilars? Now, the current regular strategies by EMA is the following. These are the new drugs with a full dossier. They can be, approved by different pathways. These are the abridged applications for generic drugs, for follow-on drugs. We have the generics, as I just told you, pretty straightforward and established for a long time. But we have the biosimilars here for the proteins, and not for non-biological drugs, but for biological drugs. So there is a possibility to have hybrid applications, which are not fully generic, but they use some data from the originator drugs, plus they have to do something else, like a clinical study, for instance, little more than for the generic applications. And the EMA thinks today that these nanosimilars, if you want, may be approved by this pathway. Up till now, they have done this only by the generic pathway, these nanosimilars, if you want, except for one single product, which was approved by the hybrid application here. Now, if you look at the FDA, they are talking not of nanosimilars, but of complex generic drug products. Presented this at a conference in 2018. And here you see all these properties that they ask you to look at. So what we call the critical quality attributes of these formulations, concerning the whole particle for the stoichiometric ratios of iron, free and bound carbohydrate, and so forth, molecular weight distribution, particle size distribution, the iron core, crystallinity, etc., carbohydrate shell, and so forth, and they give you some examples of common tests that may be used. They also tell you about impurity considerations for these iron sucrose particles, looking at low molecular weight iron and free iron species, labile iron again, where they suggest the bleomycin assay, which we found out doesn't apply. Forget the bleomycin assay. You cannot use this assay to determine labile iron. And we told this to FDA, and I think they corrected it now. The iron core, the carbohydrate shell, all this impurity consideration have to be taken into account to characterize your originator iron sucrose product, but also your follow-on, your similar product of iron sucrose. Now, what we did in my lab, we looked at this. We got a couple of batches from the originator and a couple of follow-ons, or iron sucrose similar, or ISS, from China. We bought them in the pharmacy. They're on the market there. They're supposed to be generic. Now, what we did, we looked at these critical quality attributes, different properties of these nanoparticles in terms of size, in terms of dissolution kinetics, in terms of labile iron, and so forth, and charge, surface charge. And we looked at differences. Can we find differences between the originator drug and the follow-on drugs, the similar drugs? So are these really generic or not? And what we found is significant and very significant differences between the originator and the follow-on drugs, the similar drugs. So for me, this is not generic. These are maybe similar, but they are different. And I will show you. We did determination, for instance, of labile iron. We ran, as I said, through in total 7 assays. One is not mentioned here, which I will mention afterwards. You see that we took a lot of time to determine this labile iron. So nanoparticles and then the iron that is leaking out, which is then giving the side effects. And we saw that, for this bleomycin assay, we could not prove linearity. We had side reactions. There is toxicity of the bleomycin. There's an improved version of the bleomycin assay with ethidium bromide. Same thing, linearity not proven. We had the batophenantrolene assay and this bipyridyl assay. We could not prove linearity, or the results were not reproducible. So we went to 2 others, Comazurol B and Furazine, and here, finally, we could prove linearity of the assays, and we had reproducible results. We also did a validation according to ICH guidelines, and we could find this. However, this here is a mild method, so we think that the concentration of labile iron we detect is underestimated, while this is a rather harsh method, where we think that one of the steps here, the reduction agent that needs to be applied in the assay, dissolves some of the iron out of the core, so it's overestimating the concentration of labile iron. But taking this into account, we have reproducible results, which are shown here. For instance, we have the originator drug here, the Venofer, no difference in between. We had the others, the so-called generics. You see quite some differences and very significant differences between these drugs here and also in between batches sometimes, which shows you that the production process of these nanomedicines need to be tightly controlled in order to avoid batch-to-batch variability. We also did a further assay, which gave us the concentration of IN3 and IN2 in these formulations. For this drug here, you see there's about 50-50, whereas for other drugs, this ratio was a little different. Sometimes it was always 2-to-1. So here again, you see quite significant differences in between these drugs that's supposed to be generic. Then we looked at dissolution. So once you introduce the nanoparticles into the bloodstream, how much time does it take for 50% of dissolution? We tested that in human serum, actually. And here we saw that for the first drug here, we had about 19 minutes, plus minus 1 minute, for half of the particles to be dissolved. For the others, the ISSA was much faster, so there's a much more rapid dissolution, which may give you more labile iron in the blood. Whereas the others, this one is okay. However, for this one, you saw more variability in the dissolution time. So I cannot tell you, I can make some suggestions whether one or the other is, in terms of clinical efficacy, better or more safe. However, to connect these parameters in the end to the clinical outcome is somewhat difficult. Here you see also the charge, the surface charge we measured, no difference in between the batches of this one drug here. There were large differences, batch-to-batch variabilities for this drug, and you see here as well. So there are differences, and the quality in terms of the inter-batch variability sometimes is a little strange. Now, I have just to introduce another concept. I think that's the third concept now. If you look at what happens if you introduce a nanoparticle into the bloodstream, this particle doesn't stay alone, right? So it forms a biomolecular corona around the particle. So there are like proteins and other stuff attaching to the surface. This is Mr. Vroman, who had drawn this concept some time ago. So what is basically this theory, this effect basically says is that after a while, and according to the surface properties of these nanoparticles, you get an absorption of different proteins to the surface, which then will make up this decoration with proteins, this corona, protein or biomolecular corona, right? Now we looked at this for our nanoparticles there that we had for the iron sucrose. Now if you consider albumin, which is the most abundant protein in the bloodstream, this is about 3.8 times 15 nanometers in size. Our particles are 8 nanometers in size. This is drawn to scale. Now, if you consider that parts of the complement of the immune system are up to 400 kilodaltons, IgM, for instance, almost 1 million Dalton, much bigger than these particles. Now, I think we have, for these very small particles, we have to revisit the Vroman effect, because what are you doing? Are you opsonizing these particles, or are you decorating these big molecules, these big proteins, with a number of particles? And what is, after that, the effect that you see in terms of efficacy, in terms of toxicity, if you change the surface parameters of these particles or the size of these particles? And there you have maybe again a difference between the different drugs that I just showed. Refer to Shakespeare's Hamlet. There's a quote from Shakespeare's Hamlet, is that we know what we are, but not we may be. We cannot look into the future. And this applies also as such to the nanoparticles. I told you that we can, to a certain extent, characterize our nanoparticles which we are using in the lab on the shelf. We can measure the size distribution, zeta potential. You have it. We can even look at the structure of the surface now with very advanced method. But once we put this into a biological environment, there's this formation of the biomolecular corona. So this synthetic identity that the particle had in the lab on our shelf suddenly has a biological identity which is characterized by the stuff that's adhering to its surface. And this biological identity will have an influence on the physiological response, will have an influence on the pharmacokinetics and pharmacodynamics of these nanoparticles. So the big question, which I will come back at the very end of my presentation, the big question is, can we design these nanoparticles, the synthetic identity of nanoparticles, in order to obtain a certain biological identity? So can we design this biological identity in order to obtain a certain physiological response? That's a big question. And I've mentioned here, can you design the biological identity of a nanoparticle? Can you determine a physiological response? Now, what we are doing currently in a project with different partners, we're looking at the simulation in silico, if you want, of the interaction of a molecule, transferrin, it's also present in the bloodstream, with particle surfaces that we can design. We modify these particle surfaces so that we modify the synthetic identity to increase specific binding of transferrin, to change a little bit the biological identity. So our idea would be to, if we introduce this modified particle into the bloodstream, you get a little bit more absorption of transferrin, or a preferred absorption of transferrin to the particle surface, which then could lead to an interaction with the transferrin receptor, which is expressed at the blood-brain barrier. So maybe we can use that in order to target the brain and get some nanoparticles into the brain. So, and then have the physiological response. That's the theory here. You see this transferrin swirling around here happily. And then what we do is we increase this interaction by modifying the surface here of these superpair magnetic iron oxide nanoparticles. So also rather small, like 100 nanometers in size. And so we get after that, when this transferrin is adsorbed to the surface, it may have an interaction with the transferrin receptor. However, what we also look at is, do we really get this conformation or this orientation of the transferrin so it's still able to bind to the receptor? Or do we get something like this, where the transferrin is lying flat on the surface of these nanoparticles and can no longer interact with the receptor? And this is something we are simulating in cooperation with the University of Southern Switzerland. And our colleague there, Tommaso, he is doing the simulations, and he uses the Lake of Lugano, which you see here, to cool down his CPUs. That's how much calculating power is needed to execute these simulations. After this, one more thing. I talked to you about the PK of these iron sucrose. And this is all after what's happening after the particles have been introduced into the bloodstream, right? After the infusion. Now, I'm a pharmacist, and I said my brother is a nurse. So what about the clinical day-to-day life? How does this work? Now, usually for these iron drugs that are infused into the patient, what the nurse does, she takes the dose, injects it into an infusion bag of saline, and you have a line, an IV line into the patient, and voila, your dose is delivered to the patient. Now, what if something would happen to your drug while being in the infusion bag? We did that. We looked at differences in terms of between different products when we injected them into a standard infusion bag that's used at the hospital just across the street. We injected that. We saw problems with viscosity for this product here that you see, and mixing problems we had. And we had seen differences in colors between two different drugs here that we also applied. You probably can see this, but there was, when we looked at this by eye, we saw differences in color. So already there's something happening in the infusion bag which is different from each other. So different products who are supposed to be generic behave differently in this environment. So real-world experience that is getting more and more recognition by the FDA in the whole approval process. As I said, to link these critical quality attributes, these properties of the formulations of these drugs to the outcome is somewhat difficult. However, what you can see, what you can have is some observations. Now, here I show you an observation of 658 postpartum or gynecology surgery patients, which received either the iron sucrose originator drug or the iron sucrose similar drug. And we're looking here at injection site reactions, so inflammation at the site of infusion there. For the originator here, we have 1.8%, so not much. However, when they applied the iron sucrose similar here, 13, there was an increase to 6.2%, almost 3 times or more the injection site reactions in these patients. Now, what does a nurse do? What does a medical doctor do if he sees, oh, if I inject this or if this is infused at this concentration, I get injection site reactions, so better dilute it. So they diluted this, but they got even higher side effects, because by diluting it, you destabilized the formulation. You had a better dissolution of your drugs already in the infusion bag, and that caused the injection site reaction, or the increase in injection site reactions. So that teaches us 2 things. Nanomedicines are different than other drugs. They're very complex, and they're different from small molecular weight drugs, also in their handling. And the medical personnel in the hospital who applies these drugs needs to be trained to avoid these increase in side effects. Now, there's another study. That was a retroactive study by a friend of mine, Jacques Rottenbourg, in a hospital in Paris, France. He had a couple of patients here that had to undergo hemodialysis because they had chronic renal failure, chronic kidney disease, so they had to be put on hemodialysis, which then reduced their red blood cell count, and therefore they had to receive iron sucrose and this erythropoietin in order to stimulate the synthesis of red blood cells. Now, during the first phase, everything was peachy. These are the hemoglobin counts, which have to be between here and here, so it was in a good range for these patients. They had to give a certain concentration dose of erythropoietin and a certain dose of the iron sucrose. Now, something happened after a couple of weeks. You suddenly saw, my friend there, Jacques, saw that the hemoglobin counts were way beyond the ideal threshold here, the therapeutic threshold. They had to give much more erythropoietin, 12% or almost 13% more to these patients in order to go back up to the hemoglobin levels here at this point. And they had to increase the dose of IV iron. Now, what happened was that, the technical director of this hospital saw the costs for this originator drug, which were higher than for the similar or for a so-called generic drug. So he told his guys, use this generic drugs because it has the same dose, same effect, so use that. However, what you see here is that at the same dose, this perceived generic drugs was not acting generically at all, but it showed some lower effects, lower side effects, and then you had to increase the dose here. So not generic at all. And excuse me, I have a certain I take pride in my being a pharmacist, and for me, these effects are not generic. This is dangerous for the patients. This was, as I said, a retrospective study. There have been 2 more studies on this prospectively, and they came to the same results. So there's something going on that is not generic. So we have to take care. A short excursion, we also did some, because we were working with iron products there, we also looked at iron products that can be taken orally as tablets. Sometimes people do that still. And we looked at whether there are also differences with these products. And we looked at all these drugs here. You see 2 batches of Multifer, 1 batch of Enrich, Fergluron, Hemafer, 2 batches, and 2 batches of iron 3. We got this from the pharmacy. We measured the pH and we measured the iron content. For the Multifer here, you have 5.87, 6 around this. Enrich was a little bit lower. Ferroglucone was 6.2, a little bit higher. You saw some variation here, but not too much. However, when we came to the iron 3 batch, we saw 2 things. One, the pH was much lower than for the others. It was closer to 5 than to 6. And we saw 200% of the declared dose, double the content of the declared dose in this product. So again, these are not generic. They are different. And one has to be careful. These are also nanoparticulate formulations, of course, that are taken up through the gut. However, you see here differences, quite significant differences between the different products. We also looked at the Z average, so the size of these particles. Here, the first one was about 10, a little bit smaller than 10 nanometers. These were already a little bit higher, and our iron 3 was much higher with a high size distribution on top. So there is, from a physicochemical standpoint, there are differences here. Now, coming back to the regulatory strategies, we think in the common framework, the framework that exists today with EMA, I think generic application does not apply, because I showed you significant differences, both in the physicochemical characterization as well as in the clinical outcomes. They are different. These are not generic. And for the biosimilar application, you can't, because these are not biological drugs. So the only thing that's left to use is the hybrid application. As I said, the current status of discussion now with the EMA is on this stage where we say, okay, hybrid. If we don't introduce a nanosimilar strategy, we could probably go for the hybrid application. So to conclude, well, I've talked for a long time. So to conclude, nanomedicines offer new treatment approaches. Cancer, I talked about cancer, I talked about infectious diseases, where we are involved in looking into this. They are complex systems in contrast to maybe small molecular weight drugs. That's why they are difficult to prepare. You have to really control the preparation and manufacturing process. And they're difficult to characterize. You have to know what you're looking at and which parameters you have to characterize. They have physicochemical properties, so-called critical quality attributes, which have an impact on their efficacy and safety. However, the links between these properties and the efficacy safety are not fully established at this point. They are only observational studies at this point. These nanomedicines need appropriate formulations and patient application, which means we have to train the medical staff, inform them of the characteristics of these nanomedicines. And like complex medicinal products, biological origin, they need an appropriate regulatory strategy, because, as I tried to show, this generic approach is not sufficient. And I am a member of a non-biological complex drugs group that tries to interact. It's a non-for-profit group, interest group, that tries to interact with EMA, with FDA, with the pharmacopoeias, in order to get this into their brain, that these things are not equivalent or similar, that they are similar but not generic. Now, in the year 2029, this is my perspective, not only because I will be retired by then and I will drive off on my Harley into the sunset, But in 2029, I'm sure that we will have understood which properties of these nanoparticles do influence their PK, PD, and tox profiles. And moreover, we will be able to engineer nanomedicines, their physicochemical image, their profile, accordingly in order to achieve true drug targeting. Is this science fiction? A little bit. However, you see more and more of these publications popping up that go in this direction. There was one from 2015, Nature Nanotech, mapping protein binding sites, that goes into the direction. Nanomedicine design, specifically recruit bind native transferrin in a controlled orientation, goes as a step before what we do. And you have to exploit the properties of the protein corona in order to get the design of the nanoparticles to achieve true targeting. And with this, I'd like to acknowledge all these institutions we are working together with. I'd like to thank you for your attention. If you have any questions beyond this webinar, please contact me through my email or follow me on LinkedIn and contact me on LinkedIn. And I wish everybody a very nice summer and rest of the day. And I'm open for questions if there are any. Thank you very much for your attention.
Thank you, Dr. Gerrit Borchard. Your
Yes.
presentation, you had a wonderful presentation.
Thank you very much.
On behalf of our management, hereby I express my hope you and your management for giving such an informative, elaborated presentation about the small-scale lab challenges of nano particles. So, from seeds of intelligence to the student. I hope this interaction will motivate the students and encourage them in active learning of forthcoming days.
Okay.
Professor, in future also, let's have a good collaboration for the development betterment for our students' career, Professor. Request on behalf of our management. I'm here to request you to, in future also, let's have a good collaboration.
Sure.
And betterment of our students' career, Professor.
Sure. I'll be happy to.
Yeah, another one request, Professor. Whenever would like to plan to India in future, please let me know.
Yes, I haven't made any travel plans. I'm grounded as everybody else for now, but I would be very much looking forward to come back to India a seventh time and to visit you there in Tamil Nadu. I would be honored to take this opportunity. Thank you very much.
Sure. Welcome. Any questions? Students, any questions?
As I mentioned, you can also write to me and set this in writing to me, and I try to answer the questions.
Yes. Andu, sir?
Yes, sir.
Any questions, sir? In chat box.
Sir?
Yes?
Sir, there is a question, sir.
Yeah.
Sir, last 30 to 40 years, we are seeing a lot of novel drug delivery systems. But when you are coming to the commercialization, still a lot of people are using the conventional products. In which area that the novel drug delivery system are lagging behind, sir?
So if I understood your question correctly, the connection is very bad. But if I understood correctly, you're asking for, what comes after that. We have a lot of concepts in development, but to turn them into real drugs, that is still a problem when it comes to manufacturing procedures?
Yes, sir, definitely.
Okay. That's a very good question. And many companies are facing this. Actually, we organized a a session on this at the upcoming UFEPS conference next year that had to be postponed to next year, with the contribution of somebody from AstraZeneca. They're facing this as well. So we can all make our particles in the lab. However, then the scale up is a little bit difficult. What I think is we need to precisely control the process. Like for these iron sucrose nanoparticles, people have that under control, but it took them a long time. We have to identify the in-process parameters. Is it size, charge, and so forth, in order to be able to produce and manufacture these particles with the same, under the same conditions and with the same properties in the end. Now, what we have done in this regard is during the pandemic now, where we did our work on the vaccine, we did some liposomal formulations of vaccines. There we used a equipment, a microfluidic equipment, which gave us quite reproducible results in terms of size and zeta potential and so forth of our liposomal formulations. Now, this was a medium lab scale, so it was not the smallest volume we could produce, but in between. And I understand that such equipment is also available, for instance, for large-scale production of these liposomes, for instance, but also for polymer nanoparticles. So my bet, if I could wager a bet right now, is to have this technology more developed, like microfluidic technology, in order to have to guarantee the quality of these products in the end. I think that would be my thoughts how to go forward. And I know that some of the companies are looking into this way as well.
Thank you, sir.
You're welcome.
Sir, and also, like you have told that similars are not similar each other, like that you have told, sir, like for iron sucrose combination, like that. So generally the generic products are most therapeutic equivalent or chemically equivalent, and they are going to produce same kind of pharmacological activity.
Yeah.
But according to your studies, it shows there are a lot of variation in their pharmacological activity. So it is applicable to the particular product only, or mostly it was the case, sir?
Yeah, I'm sorry, I did not understand your question completely. Were you referring to the electrokinetic mobility?
No, sir. The iron sucrose similars.
Similars, yes.
Yeah, similars. So you have told they are not therapeutically equivalent. There are a lot of variation in their pharmacological activity. So it was the case for all the drugs or for a particular drug only, sir?
So we saw differences in between the drugs we were looking at. That's the thing I can answer for the drugs we were looking at. I know when you talk to the guys who developed liposomal doxorubicin, for instance, the inventor said to me once that, you know, once you change something in the composition of a liposomal formulation, the whole thing falls apart. You have a very different profile afterwards. We have seen this for liposomal doxorubicin. That was where a company produced generic liposomal doxorubicin for the American market, and they saw differences there as well in the head-to-head study. For the European market, they saw a difference in the head-to-head study with the originator. So they could not show the similarity to the product they had before. So yes, devil's in the details. So you have to really control your production process, and there you have the same problems then as the biosimilar, the protein guys have. The production process of the originator drug is unknown. It's not public. It's also not patented, so you do not know what these companies, the originator companies, did to produce their drugs. The same applies for nanomedicines as well. So you basically have to create your own process, which then could give you, in the end, different results. I don't know whether I answered your question.
Yes, sir. Thank you. Any other questions?
I see that there are 132 persons on this webinar. I'm honored.
No, sir. There are 160 participants, sir.
Whoa, okay. Now I'm nervous.
No, and people are watching through YouTube also, sir.
Oh, okay. So
Yes,
is this
sir.
recorded?
Yes, sir. Is this recorded, sir? We will send you through mail,
Oh,
sir, that link.
thank you very much. Thank you.
Okay, sir. I think there is no other question also, sir. So, shall we conclude, sir?
Yes, it's up to you. So, as I said, I welcome any question you may have. Please send this to my personal email there, and I try to answer them as quickly as possible.
Okay, sir. Thank you, sir. Thank you for your wonderful presentation.
Thank
We
you
have
very
a
much
nice
for
time.
inviting me.
Thank you for sharing your technology advancements with us. Thank you,
Thank you
sir.
very much. Thank you.